Lymphocyte development occurs in specialized environments and is regulated by the somatic rearrangement of the antigen receptor genes. Certain basic principles apply to the process by which precursor cells develop into committed B or T cells expressing antigen-specific receptors, the immunoglobulins and T cell receptors respectively. In both humans and mice, this process of lymphocyte differentiation occurs in ordered stages, and these are marked by successive steps in the rearrangement of the antigen receptor genes and expression of their protein products, as well as by changes in the expression of other cell surface and intracellular proteins. The successful execution of this intrinsic developmental program requires signals from the specialized microenvironments in which lymphocytes develop: bone marrow and fetal liver for B cells and thymus for T cells. These tissues provide a network of specialized non-lymphoid stromal cells that interact intimately with the developing lymphocytes, providing signals through secreted growth factors and cell surface molecules that bind receptors on the lymphocyte precursor cells. The antigen specificity of each individual lymphocyte is determined through the assembly of V, D, and J gene segments to generate V genes encoding the antigen receptor variable region. The expression of a complete antigen receptor requires the successful rearrangement of two different genetic loci in order to produce the protein chains of the antigen receptor, the heavy and light chains of immunoglobulins, or the alpha and beta chains, or delta and gamma chains, of the T cell receptor. Not all gene rearrangement events are successful, however. Because of the imprecision in the recombination process, not all rearrangements produce a complete in-frame DNA sequence that can be translated into protein. The successful assembly of a V region is monitored for each locus and defines the different stages of lymphocyte development. A successful gene rearrangement, termed a productive rearrangement, leads to the synthesis of the protein product. This is the signal for the cell to progress to the next stage of development. In the case of B cells, for example, the assembly of an in-frame heavy chain VDJ sequence leads to expression of the heavy chain. This is sensed by the cell as a signal to stop heavy chain locus rearrangement, to divide several times, and then to commence rearrangement of a light chain locus. Unsuccessful rearrangements that do not result in a protein are termed non-productive rearrangements, and if no further rearrangement can occur to rescue the situation, the lymphocyte dies. To survive these first stages of development, therefore, B cells must make a productive rearrangement at the immunoglobulin heavy chain locus and at one of the two light chain loci, kappa or lambda. T cells must make productive rearrangements at either the alpha chain locus and the beta chain locus, which produces the alpha beta T cell, or at the gamma chain and delta chain loci, which produces a gamma delta T cell. Those cells that fail to make the necessary productive rearrangements die in situ by apoptosis. The events that lead to a B cell with an immunoglobulin B cell receptor and T cell with an alpha beta T cell receptor have a very close correspondence. Only one gene locus is rearranged at a time, and the loci are each rearranged in a fixed sequence. Both B cells and T cells rearrange the locus that contains D gene segments first. In the case of B cells, this is the immunoglobulin heavy chain locus. For T cells, it is the T cell receptor beta chain locus. Only if a productive rearrangement is made do developing B cells go on to rearrange a light chain locus, and T cells go on to rearrange the alpha chain locus. And that's lymphocyte development occurs in specialized environments and is regulated by the somatic rearrangement of the antigen receptor genes. Have a great day.